Welcome to Europa XP OmicsCloud, your home for data management, expression analysis, and sharing. OmicsCloud supports multiple technologies, but the basic data workflow is the same for every technology. Import files, create samples, create study, analyze data, and share. Today we're using the nCounter profiling analysis module, which is licensed on this account. OmicsCloud also supports generic expression matrices, microarrays, RNA-seq, mass spec, single-cell RNA-seq, and Olink targeted profiling. You can request an upgrade to add additional modules. And if you get stuck along the way, you can use AI Mentor, open the nCounter user manual, or contact support, all from this tab. In the Files tab, click Add New, File Upload, and select your RCC files. Tag them so they are easy to find later. Next, repeat the process and upload your probe annotation CSV in the same way, and set the type to probe annotation. If you forget to add tags, the system will remind you. Each RCC file gets an automatic QC report. 4 metrics, pass or fail at nSolver standards. Keep the files that pass and drop which fails. You can use arrows to switch to the next file. Now we will create a sample. Filter the Files tab by the RCC file type. Select the filtered files and click the Create Samples button. Enter a name, ensure the toggle to create a study is selected, and select your probe annotation file here as well, and click the Create Samples button. This one step will take you from raw data to a read-ready study. In the Samples tab, you can view your sample by clicking on it. Double-click to open it and see a matrix of your data. This is a good way to ensure all your data made it in. Now, click the Studies tab and open your study. When you first open the study, OmicsCloud asks for sample annotations. Unique sample ID is required. Batch and experimental groups like treatment are optional, but also useful. One thing to watch: if your CSV already contains unique sample ID or batch columns, delete the empty ones in OmicsCloud first. Otherwise, the merge appends a number and you will end up with duplicates. Delete the columns if needed, and upload your RCC annotation file. Choose a column name to match. In this case, I'm selecting RCC ID and sample name. Select the annotation columns to add, check your match statistics, and press Apply. If your annotations are in multiple files, you could now repeat the process until all the annotations are loaded. Then, save changes. For a standard analysis, press the orange plus sign and select nCounter Data Analysis Pipeline and continue. Provide a name for the output dataset, choose to run the analysis pipeline, and select your comparison variables. Then press Run. Click on the Results data brick to see the results. You will get the full Bruker pipeline, including QC, normalization, differential expression, pathway scoring, and cell type profiling. All of these results are available to download as CSV or SVG files. This is your one-click nCounter analysis data pathway. Now I will take you through the interactive analysis path. The pipeline produces normalized data as its own dataset. This is your starting point for deeper interactive analysis. Click on the orange normalized dataset to check for normalization results by examining box plots or comparisons, including all by correlations. This is a good way to look for outliers and check normalization quality. Before we can analyze data using a custom pipeline, we want to create column sets using annotations that we uploaded in Annotation Manager. For this example, I will create a column set for tumor grade and make a column set for that. Next, I will make another column set for tumor subtype. When done, I can sort my sample by one or both of these sets. Now let's explore the data with the PCA plot. Running the PCA analysis on the orange normalized dataset in the upper left. While that is running, I will also run a cluster analysis. Click on the blue cluster dataset to see the results. For a cluster, you can zoom in and out and select the columns and rows. For PCA, I'll color my points by subtype and check to see if my samples separate into nice clusters. Zoom and rotate to see the cluster separation. Hovering over a point will give me information about that particular sample. This is a quick way to check if my clusters make biological sense. For full control over differential expression, run a t-test directly on the normalized dataset. Here, I will select tumor grade as my comparison group, and I'm going to compare normal versus grade 1. I will also run a gene set enrichment analysis to look at pathway comparisons. I'll repeat the process for grade 2 and for grade 3. Click on the purple results dataset to see the histogram of results and also to look at a volcano plot. Select the gene sets of interest using cutoff sliders, or simply drag a box around genes of interest. Click on any dot to see the gene expression across your groups. Non-parametric tests, linear models, and covariates live in the same statistical test menu. Run ANOVA to compare changes across multiple groups. Europa XP ANOVA Explorer offers an easy way to investigate changes in genes across the whole experiment. Select individual genes to see their changes. View heat maps, top results, see the effect plot, and look at QQ plots and other diagnostics. Now let's view the results of GSCA. First, I'll review the normal versus grade 3. Because nCounter is targeted, we set a minimum number of genes per pathway in the t-test setup, so your panel is represented. Click any enriched pathway for its running score and leading edge genes. The volcano plot shows in red which genes are contributing to the score. No exporting and no R is needed. Now compare multiple pathway comparisons to find pathways that stand out. These are just some of the functions of nCounter analysis in Europa XP OmicsCloud. You get one-click analysis pipeline when you want it, and full interactive exploration when you need it. The same platform also handles other omics technologies when you are ready, including cross-omics analyses. Visit europadx.com to get started. Thanks for watching.